name is Nikolai, and today we'll be talking about the clinical potential of induced pluripotent stem cells, also known as IPS. Now, the first time I was introduced to IPS, I was like, what the heck is that? Sort of how some of you may be feeling right now. But if we break these words down, I think we'll have a better understanding of what IPS really is. The first word is induced, a form of the verb induce, which means to bring about or give rise to. So logically, the next thing to ask is, what is being brought about? And this is answered by the next three words. So in biology, pluripotent and stem cells are essentially synonyms, both referring to unspecialized cells that are capable of differentiating into multiple cell types. So in essence, an induced pluripotent stem cell is just a cell where you've brought about the ability to differentiate into multiple cell types. And this is significant because, as you all know, a majority of cells in our body are specialized, meaning they've been designated to perform a specific job to keep our body healthy. For example, red blood cells are specialized cells with the job of transporting oxygen. However, these red blood cells weren't always specialized. Like all cells, they started out as regular unspecialized stem cells. Think of stem cells as cells that are still in training. They haven't been assigned a job yet. But as soon as they are assigned a job, let's say to deliver oxygen, they become specialized in a process known as differentiation. Now, differentiation had always been considered a one-way process. We knew stem cells could differentiate to become specialized, but we never thought that specialized cells could ever undifferentiate to become stem cells once more. At least not until 2012, when scientists from Japan won the Nobel Prize for the discovery of 4 genes that are responsible for pluripotency. And these genes are OCT4, SOX2, CLIF4, and c-MYC, also termed the Yamanaka factors, named after their namesake, Shinya Yamanaka. And although these genes are present in all differentiated adult body cells, such as the skin cells shown here, they're usually turned off. And the scientists wanted to see if turning on the Yamanaka genes would allow the differentiated cells to go back in time and become undifferentiated. So, to reactivate the genes, the scientists added additional copies of the Yamanaka factors directly into the genome of the cells in a process known as viral transduction. They found that their hypothesis was correct, and that reactivating the Yamanaka genes by adding additional copies of the Yamanaka factors allowed the previously differentiated skin cells to become undifferentiated IPS cells. And thus they were able to induce pluripotency within these cells, which is where the name IPS actually came from. So if you're confused, don't worry. Here's a simple analogy. In the first stage, the undifferentiated cell is performing its designated job. So if it's a red blood cell, well, let's say it would be differentiated, it would be delivering oxygen. But by adding the Yamanaka factors, you're essentially telling the cell, hey, we don't want you to be delivering oxygen anymore. So these cells then become an IPS, which is when the cells are just chilling, not doing any specific job because they don't have any instructions. But as soon as they get new instructions, through the addition of cell-specific genes, cells become specialized once again into the cell type that you gave them instructions for. So for example, if you wanted to create neurons from IPS cells, all you would have to do is add these specific instructions or genes for neurons. And the scientists were actually so shocked by the results that they conducted a sort of proof of concept experiment, where they specifically designed a cell to glow green when it's become a stem cell. So these cells are shown here in their original differentiated state with a nice gray hue. Then as soon as they added the Yamanaka factors, the cells miraculously turned bright green, signifying that the Yamanaka factors had indeed induced a pluripotent stem cell state and activated stem cell genes. So after this breakthrough, scientists began work on harnessing the immense potential of IPS in a clinical setting. The discovery of IPS and the Yamanaka factors makes it possible for doctors to isolate, for example, skin cells from a patient, turn those skin cells into undifferentiated IPS cells, and redifferentiate those IPS cells into any functional cell that they may desire, including neurons for the brain, alveoli for the lung, hepatocytes for the liver, anything. So these functional cells can then be safely transplanted back into the patient, thus curing any disease they might have had, whether it be Parkinson's, diabetes, even Alzheimer's. And best of all, these cells have the identical genome as every other cell in the patient's body, meaning there is no potential for rejection, which may happen in the case of whole lung transplantation or whole organ transplantation. So in essence, IPS cells could hold the key to treating every human disease that currently exists, potentially even halting the process of aging itself. So thank you for watching my presentation. I hope you learned as much as I did.